Today, we're going to explore the fascinating world of machine learning and its applications in medical healthcare. Lecture focuses on the relevance of bioinformatics applications in the discovery of vaccine candidates and potential drugs for COVID-19 treatment. Now we're diving into how bioinformatics helps us fast-track finding vaccines and drugs for COVID-19. Think of it as a data treasure hunt where we decode genetic info to outsmart diseases with all the data at our fingertips. We're getting quicker at spotting the pathogens, understanding diseases, and inventing new tests. And it's amazing how we use this data to pinpoint the proteins that let viruses invade our cells. Bioinformatics has been a huge boost in our search for COVID-19 solutions. We'll explore how these tech tools have been a big help and how they'll continue to revolutionize medical discoveries. First, let us start with the application of bioinformatics in vaccine design for COVID-19 treatment. There are three applications, which are reverse vaccinology, immune informatics, and structural vaccinology. First, reverse vaccinology uses to identify novel antigens by analyzing an organism's genomic information. It is a cost-effective process. Other than that, it also uses to identify target allergens using information on the genetic makeup of the pathogens. Second, immune informatics uses for analyzing complete information on an organism's immunomics. It also uses generated data to make prediction of immune responses against specific molecules. Lastly, structural vaccinology uses bioinformatics in predicting vaccine candidates for SARS-CoV-2 and identification of structurally stable, safe, and effective peptides as vaccine candidates. Let us continue with the steps that is required in immunoinformatics. These are the steps for immunoinformatics. Let us dive into the details. For data retrieval, we need to start by collecting proteomic data relevant to the vaccine design, then use this data to perform computational analyses to predict immune responses. From the data, it will be used to test. First, antigenicity, where we check if the peptides can induce an immune response. Second, allergenicity, where we want to ensure the peptides do not cause any allergic reaction. Third, peptide toxicity, where we want to verify the peptides are not toxic to human cells. And lastly, epitopic conservancy, where we want to confirm the epitopes are conserved across different strains or variants. At the end of the immunoinformatics, vaccine need to be validated using computer simulations. Now we are done with the steps. Right now, I will be explaining about the process of immunoinformatics. The process starts with data retrieval, data editing, physicochemical testing, epitope prediction, peptide promiscuity prediction, and end with quality checks. First, data retrieval is achieved by obtaining nucleotide or proteomic data from a biorepository. Second, editing the data using bioediting tools and remove deletion sites manually. Third, physicochemical testing is done to test sequences for parameter. As an example, amino acid composition, half-life index, solubility, and elemental contents. Fourth, by using machine learning tools, prediction of B and T cells epitopes are done. Fifth, peptide promiscuity prediction is done based on MHC1 and MHC2 allele restrictions. Lastly, we end the process with performing quality checks. That's all for the process part in immunoinformatics.
Next, moving on to the emergence of the new SARS-CoV-2 viral strain, which is the VUI20201201. The new strain is characterized by a total of 17 mutations and emerged in the United Kingdom in December 2020. The most prominent mutation is the N501Y mutation in the spike protein. This mutation enhances viruses' ability to bind to human ACE2 receptor. This potentially increases its infectivity and rate of spread. Next, this strain has impacted the vaccine development, in which the increased infectivity of new viral strains speeds up the transmission rates and potential decrease in efficacy of existing vaccines. Additionally, the development of bioinformatics tools and computational methods have aided in monitoring and analyzing viral mutations, quick adaptation to new data, and in informing vaccine design. Moving on to the limitations of immunoinformatics for predicting vaccine candidates. The major issue is the inappropriate animal models. Selecting the right animal models is critical, as some SARS-CoV vaccines failed in trials, either not providing full immunity or causing adverse effects, like lung inflammation in mice. Next, reverse vaccinology issue. Reverse vaccinology targets mainly proteins and overlooks other important biomolecules like polysaccharides, which can enhance immune responses. Furthermore, epitope prediction challenges in which many predicted epitopes are not accessible to antibodies in vivo, despite promising in silico results, leading to potential vaccine failures. Lastly, MHC class I molecule diversity that complicates accurate epitope prediction, impacting vaccine design. Now, let's look at the role of bioinformatics in drug design for COVID-19 treatment. Firstly, bioinformatics help in the identification of drug targets like protease structure. Identifying the crystal structure of SARS-CoV-2's main protease, MPRO, facilitates drug repurposing. Next, bioinformatics also helps in the 3D structure elucidation, by aiding in drug repurposing and discovery as well as providing templates for drug design. To dive deep in drug repurposing, computational screening methods are used to screen existing drugs for potential efficacy against SARS-CoV-2. Also, network-based drug repurposing platforms are employed to identify drugs based on interaction networks and genomic sequences. Moreover, in silico methods like molecular docking for prediction of interaction with viral proteins are also utilized. Additionally, computer-aided drug design like QSAR optimization are used to predict the efficacy and optimize drug candidates. Last but not the least, bioinformatics also includes AutoDock offline program for docking studies and drug repurposing efforts. So now let's look into network bioinformatics approach in drug repurposing prediction. First of all, let's see how does it work. First, by searching the human interactome for viable drug targets. Next, identify repurposable drug candidates. Then, development of combination of actions such as selection of virus and virus-host interactions and suitable drug targets. As an example, COVEX or Coronavirus Explorer, a vital tool massively used in the SARS-CoV-2 drug prediction. Alright, now let's dive into drug target for the MPRO of SARS-CoV-2. MPRO of SARS-CoV-2 is a key enzyme in aiding viral replication and transcription. Yang et al. Identified a mechanism-based inhibitor adopting CAD and determined crystal structure of MPRO of the virus combined with identified inhibitor. Identified 6 potential compounds that inhibited the enzymatic activities of MPRO. Epsilon is one of the identified compounds that exhibit promising antiviral activity in cell-based assays. Let's look into machine learning and its applications for vaccine and drug design. Some of the computational tools used in the prediction of antigenic peptides for vaccine design are SPRINT, a sequence-based prediction of protein peptide binding sites using support vector machine, TEPI tool, a pipeline for computational prediction of T-cell epitope candidates, and MHC-PRED which predicts cytotoxic T-cell epitope. Some of the computational tools used in drug design are CHARM, short form for chemistry at Harvard Macromolecular Mechanics, Zinc Pharma, an open-source pharmacophore-based screening program. Lastly, AMBER, a suite of biomolecular simulation programs. In conclusion, global bioinformaticians responded swiftly to the COVID-19 pandemic by offering specialized instruments to further SARS-CoV-2 research for the disease's early diagnosis and treatment. Drug repurposing facilitate the identification of potential drugs via the screening of existing drugs and protein interactions, which is comparatively cheaper and more time-effective than traditional approach to drug design.